So looking at this, we can obviously see the obvious of which we all know. Weight loss significantly improves metabolic and cardiovascular health. And so someone may want the definition of metabolic and cardiovascular health. So what is metabolic and cardiovascular health? Metabolic health refers to how well your body processes food into energy and maintains internal balance, which is crucial for overall health and reducing the risk of chronic diseases. Cardiovascular health refers to the health of the heart and blood vessels. Cardiovascular health refers to the health of the heart and blood vessels. It encompasses the proper functioning of the cardiovascular system, which is crucial for overall health. Cardiovascular health is vital as it can prevent disease such as coronary heart disease, stroke, and heart failure. Maintaining good cardiovascular health involves regular exercise, a balanced diet, and monitoring blood pressure and cholesterol levels. We can see here that metabolic health and cardiovascular health are two main functions within your body that are vital when it comes to living. Metabolic health referring to digestion and how you're able to process energy. So, the remodeling of adipose tissue is central to these varied and important clinical effects. And so we know that specifically the position by which you are at in terms of body fat percentage has a huge effect upon your metabolic and cardiovascular health. So, the remodeling of this adipose tissue, significantly gaining or losing body fat, has a huge effect upon these two things. However, surprisingly little is known about the underlying mechanisms presenting a barrier to treatment advances. Here we report a spatially resolved single nucleus atlas comprising 171,247 cells from 70 people. So what is a spatially resolved single nucleus atlas? A spatially resolved single nucleus atlas is a scientific resource that combines spatial information with single nucleus RNA sequencing data to analyze the distribution and characteristics of individual cells with a tissue or organ. This type of atlas allows researchers to understand the spatial organization of cells and their functions, enabling insights into various biological processes and diseases. For example, it can be used to study distribution of specific cell types in the brain or other tissues, providing a comprehensive view of cellular architecture and function. So then, let's go a little bit more depth upon this mechanism by which we can find the function of cells. The single nucleus RNA sequencing, snRNA sequencing. This technique captures gene expression from individual nuclei of cells. Remember, nuclei of cells are specifically, you can either have one nucleus or there's some cells that have multiple nucleus. There's mononuclei and then multinucleated. Okay. So we have mononuclei cells, which has one nucleus, and then we have multinucleated cells, which have multiple nucleuses, right? And so these nucleuses contain DNA information that can be used for cellular processes and coding and whatnot. The nucleus is vital when it comes to just overall cellular response and how our body adapts to, let's say, even just specifically training, you know? This technique captures gene expression from individual nuclei of cells rather than whole cells. Why nuclei? Because in tissues like fat or brain, isolating intact cells is hard. Nuclei are more susceptible and still contain rich transcriptional data. And so I find this really interesting because what we're doing is that instead of trying to take the whole cell on its own, what we're doing is that we're grabbing the information within the cell instead in order for us to make it a little easier for us to take in information about how maybe a cell would react to a particular part within our body, such as adipose tissue. And I think this what's going to make this research paper really interesting is that what we're going to do is we're going to evolve the response via the nuclei given the RNA sequencing in relationship with adipose tissue. And that leads me to question, how is our response to losing weight, specifically via the nucleus, via the RNA sequencing, different with muscle tissue in comparison to adipose tissue? And that's something that we're probably going to be able to look at later on. So it tells us what genes are active in each cell, giving a molecular fingerprint of cell identity and function. Spatial resolve. This means data isn't just floating around in a spreadsheet, it's mapped back to the tissue's physical layout. You know where each cell was located, near a blood vessel, in a fibrotic zone, adjacent to immune cells, etc. It's like turning a list of ingredients into a full recipe with plain instructions. A compressed, comprehensive, annotated map of all the cell types and their spatial relationships within a tissue. Think of it as a Google Maps or biology. You can zoom in on neighborhoods, cell niches, traffic pattern signaling, and even construction zones, remodeling, or pathology. Why is this powerful? Traditional RNA sequencing versus spatial atlas. Cell level resolution. Traditional RNA sequencing, no. Spatial resolved atlas, yes. Spatial context, no. Yes. So basically, when it comes to being able to figure out the tissue architecture, the spatial context, the cell level resolution, the cell-cell interactions, the clinical relevance. Basically, all the information of how the cells interact via cellular processes and whatnot is able to be figured out via the spatially resolved atlas method. Go into RNA and break down what RNA really is. So RNA, complex compound of high molecular weight that functions in cellular protein synthesis and replaces DNA, deoxyribonucleic acid, as a carrier of genetic codes in some viruses. RNA consists of ribose nucleotides, nitrogenous bases appended to a ribose sugar, attached by a phosphodiester bonds, forming strands of varying lengths. The nitrogenous base in RNA are adenine, guanine, cytosine, and uracil, which replaces thymine in DNA. Okay, so we know that RNA is a complex compound, and it functions in cellular protein synthesis. And it replaces specifically DNA, deoxyribonucleic acid, as a carrier of genetic codes in some viruses. So RNA consists of ribose nucleotides, nitrogenous base appended to ribose sugar, attached by a phosphodiester bonds, forming strands of varying lengths. That the nitrogenous base in RNA are adenine, guanine, cytosine, uracil, and thymine. So let's look at these nitrogenous bases. Or adenine, an organic compound belonging to the purine family, occurring free in tea or in combined in many substances of biological importance, included in nucleic acids which govern including the nucleic acids which govern the hereditary characteristics of all cells. So what are nucleic acids? Nucleic acids are naturally occurring chemical compounds that serve as the main information carrying molecule of the cell and that directs the process of protein synthesis, thereby determining inherited characteristics of every living thing. So we know that nucleic cells are what build up RNA, and RNA is vital when it comes to protein synthesis, creation of amino acids and whatnot. So looking at here, we can see that nucleic acids are vital, naturally occurring chemical compound that is vital when it comes to the structure of RNA, in which RNA, genetic carrier after DNA that replaces DNA, is vital for protein synthesis. So let's look at here and let's look at RNA's part in protein synthesis. Protein, highly complex substance that is present in all living organisms. Protein of the great nutritional value and are directly involved in the chemical processes essential for life. The importance of proteins was recognized by the chemists in the early 19th century, including Swedish chemist Jöns Jacob Berzelius, who in 1838 coined the term protein, a word derived from the Greek proteus meaning holding first place. Proteins are species specific, that is, the proteins of one species differ from those of another species. They are also organ specific. For instance, within a single organism, muscle proteins differ from those of the brain and the liver. Protein molecule is very large compared with the molecules of sugar or salt and consists of many amino acids. There are about 20 different amino acids that occur naturally in proteins. Proteins of similar function have similar amino acid composition and sequence. Although it is not yet possible to explain all the function of a protein from its amino acid sequence, established correlations between structure and function can be attributed to the properties of the amino acids that compose proteins. Types of RNA involved in protein synthesis. mRNA carries genetic code from DNA to ribosome, contains codons, 3 base sequences that specify amino acids. Transfer RNA delivers amino acids to the ribosome, has an anticodon that pairs with mRNA codon, carries specific amino acid, and then you have the rRNA, the ribosomal RNA, structure and catalytic component ribosomes. It forms the core of ribosome structures and catalyzes peptide bond information. So seeing here, we have our messenger RNA, we have our transfer RNA, and we have our rRNA, our ribosomal RNA. And so our messenger RNA carries the genetic information. The transfer RNA delivers amino acids to the ribosome. So the mRNA and the tRNA interact with each other. The tRNA specifically carries specific amino acids to sequence with these. What's cool is that so the mRNA, the messenger RNA, carries genetic code while the transfer RNA delivers amino acids to the ribosome. And so together the mRNA and tRNA seem to work together in order for them to create a specific code. The rRNA, the ribosomal RNA, is a component of the structural and catalytic component of the ribosome, and the ribosome is what's responsible for protein synthesis. Step by step. Transcription, DNA to mRNA. In the nucleus, DNA is transcribed into mRNA. That's our messenger RNA that carries information. RNA polymerase reads the DNA template and synthesizes as a complementary RNA. So, DNA is transcribed into mRNA. RNA polymerase makes a copy that will read the codons and attach with the mRNA, and this mRNA carries genetic instructions for building a protein. So together, this mRNA and the tRNA will work together in order for us to create this protein via protein synthesis, mRNA processing, and eukaryotes. Introns are removed, exons are spliced together, a 5' and a poly 8 tail are added for stability and export. What are introns and what are exons? Introns are non-coding segments of DNA found with genes that do not code for proteins and are removed during RNA processing. Okay, what are exons? Exons are segments of DNA or RNA that code for proteins. They are the coding region of a gene that contain the information required to encode a protein. So it seems like introns are not vital when it comes to protein synthesis in comparison to the exons that carry this information and is vital when it comes to the matching of mRNA with tRNA. A 5' and a poly A2 are added for stability and export. Translation mRNA to protein occurs in the cytoplasm at the ribosome. Initiation ribosome binds mRNA at the start of the codon, usually AU. So we have adenine, which are the adenine, uracil, and guanine, I'm assuming. Elongation. tRNA molecules bring amino acids to the ribosome. Each tRNA anticodon pairs with the corresponding mRNA. RNA catalyzes the formation of peptide bonds between amino acids. So it seems like, is this tRNA separate from the... Okay, so yeah, the RNA polymerase synthesizes a complementary mRNA strand, and in result, there's a tRNA strand that comes along and catalyzes with the mRNA strand. The RNA catalyzes the formation of peptide bonds between amino acids. So ribosomal RNA. Termination. When a stop codon is reached, the newly formed polypeptide is released. And so obviously, so within this synthesization of proteins, there's going to be stop codons that are going to release proteins and allow for other proteins to be made. Why is RNA so crucial? Versatility. RNA can carry information, mRNA perform catalysis, RNA, and facilitate decoding, tRNA. Regulation. RNA levels and modifications can finally tune protein production. Evolutionary significance. RNA is thought to have preceded DNA and proteins in early life forms, hitting its foundational role. So RNA is a complex compound of high molecular weight that functions in cellular protein synthesis via being vital for being after DNA sequencing, in which mRNA and tRNA are made to match together to create proteins, and then stop via stop codons. And ribosomal RNA is vital when it comes to the creation of these proteins because they create the structure of ribosomes. Thing that we're going to look at before we continue on with the research is what is RNA and what is DNA specifically, and what are its differences? Why are we doing RNA sequencing when we're trying to evaluate the cells and its place when it comes to a cell-to-cell interaction, as well as where it's placed in our body, such as on adipose tissue or muscle tissue? DNA, deoxyribonucleic acid, and RNA, ribonucleic acid, are essential nucleic acids that play crucial roles in genetics and cellular functions. So, as we see here, traditional RNA sequencing versus spatial atlas, what we're doing is that on our first step is tissue preparation. Samples are flash frozen or fixed to preserve structure. Nuclei are isolated from tissue slices. snRNA sequencing. Each nucleus is tagged with a barcode. RNA is sequenced to reveal which genes are active. Spatial mapping. Using imaging, immunofluorescence, histology, and computational alignment, each nucleus is placed back into its original location. Algorithms stitch together molecular data with spatial coordinates. Annotation and analysis. Cell types are identified: adipocytes, macrophages, endothelial cells, etc. We obviously know adipocytes, fat cells, macrophages, and endothelial cells are vital when it comes to avoiding antigens and protecting us, such as, for example, our liver and those liver globules from taking in any sort of bad bacteria and fungi when it comes to processing red blood cells, right? Macrophages, of course, and can be pro anti-inflammatory based on the state of what your liver's at, and also comes to have a really big importance when it's just protection, our first layer of protection via antigens, reactive oxygen species, whatnot. Spatial patterns are analyzed, clustering, proximity, and gradients. Functional states: senescent, inflammatory, regenerative, are inferred from gene expression. Application: disease and physiology. Brain: map neurons, glia, and vasculature in Alzheimer's or Parkinson's. Track how inflammation spreads or how synaptic niches degenerate. Adipose tissue like in obesity. Identify which fat cells are metabolically active versus senescent. What is senescent? Okay, so senescence is the loss of a cell's power of division and growth. Adipose tissue, like identify which fat cells are metabolically active, so which fat cells are able to metabolize via transfer into energy versus the lack of proliferation and differentiation. See how immune cells infiltrate and where fibrosis occurs. Fibrosis, the production of scar tissue via inflammation. Track how weight loss reprograms specific zones. Spatial atlases review how cells influence each other, not just what they are. This is crucial for understanding feedback loops, therapeutic targeting, and disease progression. So basically what we're going to look into today, I know that was very long, but I think this gives us a really great breakdown as to what we're going to be reading, is that we're going into nucleuses. Well, first we're isolating cells, right? In this case from a specific part of tissue in our body, freezing it, and to keep its figure. And then we're using the nucleus via DNA. DNA is inside the nucleus. RNA can either be inside the nucleus or in the cytosol of the cell. And so we're using DNA via the usage of RNA, which is transcribes DNA and is important for protein synthesis. And we're reading RNA, specifically RNA sequencing, in order to transcribe information via about the cell, its relation with other cells, where it's placed, and a following of other things. Senescence via proliferation differentiation when it comes to cells via in the adipose tissue, so adipocytes. And then I'm also curious upon what is the difference in RNA sequencing via protein synthesis when it comes to, let's say, adipocytes in comparison to maybe a cell like, for example, macrophages. Obesity affects more than 1 billion people worldwide. Wow. We have 8 billion people on this planet. It's 1/8 of our planet. That's crazy. Increased adipose tissue mass, which is the defining feature of obesity, is one of the main risk factors for type 2 diabetes. Type 2 diabetes, a lack of being able to take in insulin properly via the cells, so insulin resistance, cardiovascular diseases, certain cancers, and early death. A reduction in adipose tissue mass throughout weight loss significantly improves obesity-induced comorbidities. Adipose tissues have a unique capacity to adapt their structure and functions to maintain metabolic homeostasis as energy demands change. In obesity, excess expansion limits this flexibility and induces pathological remodeling changes, notably adipocyte hypertrophy, immune cell infiltration, pro-inflammatory cytokine release, impaired angiogenesis, and fibrosis that contribute to multi-organ inflammation and insulin resistance, metabolic dysfunction, and disease. So we know here that adipose tissue has the ability to maintain homeostasis, maintain balance, and also increase via hypertrophy. So as we continue to, increase the amount of calories we're eating via caloric intake, the adipose tissue has the availability to expand via increase in fatty tissue. Therapeutic WO leads to a reduction in adipose tissue mass, systemic inflammation and insulin resistance, as well as subsequent improvements in obesity-related comorbidities. This strongly suggests that WO ameliorates adipose tissue dysfunction and its harmful physiological effects. Surprisingly little is known about the underlying mechanisms. Indeed, certain adipose tissue responses to WO, to weight loss, may be maladaptive and predisposed to weight regain. Define the cell types. Regulatory mechanisms and signaling pathways are responsible for pathological and therapeutic adipose tissue remodeling and is needed to guide therapy development for the harmful health consequences of obesity. Mapping adipose tissue remodeling dynamics. To better understand obese adipose tissue dysfunction and its reversal after weight loss, we carried out single nucleus RNA sequencing of approximately 100,000 cells for men and women with extreme obesity, before and after weight loss surgery, and from healthy lean controls. Weight loss significantly improved metabolic parameters, although not the lean baseline. We focused on abdominal subcutaneous adipose tissue because of its contribution to central obesity and its adverse metabolic effects relative to other subcutaneous adipose depots. This cohort formed the basis formed between group exploratory analysis. Our results were integrated with a further 50,000 cells nuclei from the largest published human subcutaneous adipose atlas to improve cell annotation. Spatial transcriptomics and equivalent cohorts, approximately 25,000 cells, n equals 4 per group, extended data figure 1b and extended data table 1 enabled us to orient and contextualize cell phenotypes within the organized hierarchy of healthy and dysfunctional adipose tissue. This is a graphical representation of the primary study cohort. Left, single nucleus analysis and n equals 25 obese people before and after weight loss, and n equals 24 lean people with spatial analysis, and n equals 4 people per group. And adipose tissue anatomical location is right. So it seems like here we have a lean group, and then we have, in comparison with the other group, the obese group that underwent weight loss. And so in this way, this allows us to see how a person who doesn't have obesity is in comparison to someone that has obesity that went down to a healthy rate. They captured a rich representation of the cellular, structural, and functional dynamics of subcutaneous adipose tissue niche in human weight gain and weight loss. Tissue-wide clustering and compositional analysis demonstrated the extensive immune cell, mainly macrophage, but also lymphocyte infiltration in obese adipose tissue. Obese adipose tissue also showed a deficit in mature adipocytes, suggesting increased cell death and or failure to replenish mature adipocytes. Weight loss mitigated these typical deleterious effects. Immune cell infiltration is a pathogenic feature of obese adipose tissue, but the impact of weight loss on inflammatory and remodeling is unclear. With studies indicating opposing anti and pro-inflammatory effects, we clustered myeloid cells into heterogeneous subclasses of adipose tissue macrophages and monocytes and dendritic cells. So what's going on? The increase in adipose tissue macrophages primarily comprised lipid-associated macrophages. So last time, what did we learn about when it came to the liver is that we have two types of macrophages. We have M1 and M2. M1 macrophages aren't typically the ones you want to receive. They're received from what I'm pretty sure I might be mixing them both together, but there's one where it's via anti-inflammatory signaling, but there comes to where, and I think it's M2 signaling, where it comes due to via adipose tissue surrounded by the liver, which causes the macrophages to be pro-inflammatory and end up actually worsening the condition of fibrosis. The increase in adipose tissue macrophages mean from 14% to 31%. The increase in adipose tissue macrophages mean from 14% to 31% primarily comprised lipid-associated macrophages, mature MYE2 and immature MYE3 expressional. So as we can see here, yeah, we have M2 macrophages and M3, I guess, macrophages as well. And so these associated macrophages are pro-inflammatory because they are signaled via the use of adipose tissue. In this case, macrophages that were brought up with these high-fat obese clients were M2, M3 type macrophages, expressing lysosomal lipid metabolism and metabolic activation markers. Classical monocytes, MYE5, expressed in VCAN also increased, indicating constitutive trafficking of blood. Visualization marker and gene patterns supported a differentiation continuum from monocytes to immature and then mature. Proportional analysis revealed lower fractions of tissue-resident macrophages expressing homeostatic markers. Neighborhood grabs confirmed that this represented a relative, not absolute, TMR reduction. Proliferative macrophages expressed MC1, MCP, CCL2, TRM, and LM markers supporting low-level MCP1 dependent expansion of both populations in human obesity. So it seems like here that there was less of M1 type macrophages, neighborhood, or I think tissue resident macrophages, which are ones you want when it comes to in this case anti-inflammatory, and instead more of a production of pro-inflammatory cytokines. Independent of adiposity, LIM abundance increased with metabolic dysfunction, extended data figure to be. This led us to hypothesize that LIMs might have pleiotropic adaptive and maladaptive features. LIM subclustering revealed two main subpopulations that separated on lysosomal or metabolic and inflammatory signatures. Inflammatory element, what is LIMS in this case? We'll figure it out in a second. A LIM subclustering revealed two main subpopulations that separated on lysosomal or metabolic and inflammatory signatures. Inflammatory LAMs expressed higher TLR2 and TRAM1, cooperative receptors that initiate and amplify inflammation and pathogen recognition response. In keeping with the deleterious role, inflammatory LAM members increase in obesity in association with metabolic dysfunction. Spatial and protein analysis indicated context-dependent orientation of functions with adaptive LMS aggregating at crown-like structures and inflammatory being more abundant in isolation of pairs. So it seems like right here that along with increase in obesity, along with adipose tissue, there also is a production of pro-inflammatory cells. That's what I took from this. And when it comes to, we see right here, in keeping with the deleterious role, inflammatory LAM numbers increase in obesity with association with metabolic dysfunction. Or metabolic dysfunction specific, we know that metabolic dysfunction comes with the increase of obesity, and so as we increase obesity via the increase of adipose tissue, so as well as inflammatory LAM numbers, which is what we're going to go into. What is LAM numbers specifically, and what's its importance when it comes to adipose tissue? General picture. Macrophages and fat, AT macrophages, ATMs, and lean tissue. Macrophages are mostly tissue resident TRMs that keep homeostasis. Macrophages are mostly tissue-resident TRMs that keep homeostasis. In obesity, their fraction increases a lot, from 14 to 31% of immune cells in adipose tissue. Most of this increase is lipid-associated macrophages, which is not good. Okay, so LAMS refers to lipid-associated macrophages. And so what we can assume from what we read is that the increase of lipid-associated macrophages also increases the amount of inflammatory response, increases inflammation, which we know that lipid-associated macrophages also increases fibrosis via the liver. And so we can assume just by before we even go deeper into this, that it increases the overall, I guess, dysfunction of your metabolism as well as your cardiovascular health, right? Step 2, origins and differentiation. Classical monocytes, MYE5, VChem+, come from blood and infiltrate adipose tissue. Differentiation continuum, blood monocyte, immature LAMS, mature LMS. This shows obesity drives recruitment and differentiation of macrophages into lipid handling phenotype. So what that just means is basically that through So what that just means is that there's an assistance via differentiation and proliferation. Proliferation, growth, differentiation. It's like cells given a task to do something. There's an increase in protection of this adipose tissue brought by the macrophages. Step 3, loss of resident macrophages. Tissue resident macrophages normally express LYV1, FOR2, MRC1 homeostatic genes. The relative proportion decreases in obesity, but not absolute numbers. The space gets filled with LMS and infiltrating monocytes. So it seems like the tissue-resident macrophages, the macrophages that are anti-inflammatory, get pushed away to the side, while pro-inflammatory macrophages and other sort of monocytes that come with the increase of adipose tissue comes along with adipose tissue gain via obesity. Step 4, expansion via MCP1. Some macrophages proliferate locally. They express MCP1 CCL2, which recruits more monocytes. This sustains a loop. More TRMs and LAMs get generated inside obese adipose tissue. So, adipose tissue just keeps on regenerating via the uses of macrophages, and these macrophages proliferate. And so we can just tell just by reading this is that fat tissue in general can maintain homeostasis and in fact proliferate and grow. LAMS aren't all the same. Subclustering show two main groups. LAMS21, adaptive, high in lysosomal and metabolic pathways, function help clear liquids, process debris found in crown-like structures, CLS around dead, dying adipocytes, likely a compensatory protective role. LMS2, inflammatory, is maladaptive, high inflammatory pathway. MHC2, NLRP3, inflammasome, expresses TLR2 plus TREM1, which amplify inflammation, increase in obesity and associated with metabolic dysfunction, bound alone or in pairs, not tightly clustered. So it seems like within these lipid-associated macrophages that it's actually not all that bad. And while there are some that can take on to that protective role, there are also some that can cause more inflammation. So lipid associated macrophages don't just expand with obesity. Even at the same fat mass, their abundance correlates more with metabolic dysfunction, insulin resistance, dyslipidemia. Suggests that suggests lipid associated and so this suggests that lipid associated macrophages are directly tied to metabolic disease and not just fat gain. And so we can okay, so that's pretty self-explanatory. Lipid associated macrophages can't just increase via proliferation and differentiation, but can also obviously make your metabolic health worse and make it worse for your cardiovascular disease, right? Cardiovascular health, which is honestly not a surprise, but obviously it suggests that lipid associated macrophages are directly tied to this issue that you have when it comes to your metabolic health and your cardiovascular health. So, simplified version, in lean fat macrophage triple proportion dominated by lipid associated macrophages. These come from blood monocytes that enter fat and mature. Two types of lipid associated macrophages emerge. Two types of macrophages emerge. Adaptive lipid associated macrophages. These clean up lipids, dead fat cells, possibly protective. But then we also have our inflammatory lipid associated macrophages. So in terms of lean fat, where now what is considered lean fat? Let's get that out of the way. What is considered lean fat? Oh, lean body mass. Okay. Okay, so in lean fat, macrophages keep things stable, they protect us. While in obesity, when it comes to adipose tissue specifically, macrophages tend to take over, but these are going to be lipid associated macrophages instead of macrophages via M1. And so while there are lipid associated macrophages that are good, there are also pro-inflammatory macrophages, M2 macrophages that release inflammatory signals and worsen your metabolic and cardiovascular health. So, to provide an unbiased understanding of macrophage metabolic reprogramming, we use gene expression to model metabolic flux systemically. This revealed a global activation state exclusive to obese macrophages, encompassing known and previously unrecognized metabolic changes. Specifically, we found a shift to high glycolysis, pro-inflammatory, high respiratory anti-inflammatory profile consistent with extracellular flux analysis in obese mice, corresponding changes in pentose phosphate pathway and TCA cycle, pervasive activation of cholesterol, lipid, and fatty acid synthesis and oxidation pathways, obligatory upregulation of cellular transport. So what is this TCA cycle? The TCA cycle is a series of it's a Krebs cycle. Okay. I just never knew it was responded as that way. Pervasive activation of cholesterol, lipid, and fatty acid synthesis and oxidation pathways, obligatory of upregulation of cellular transport. Taking fatty acids as an example, flux modeling uncovered significant activation of fatty acid desaturation and mitochondrial B oxidation, consistent with buffering and utilization of potential toxic microenvironmental fatty acids for energy. So we know that mitochondrial B oxidation, beta oxidation is the process by which, if I remember correctly, it's either one of the two, by which we produce an excess amount of acetyl CoA via the Krebs cycle, so we actually, I'm pretty sure via beta oxidation, we would take out these subunits of what? We take out carbon, so we take out carbon subunits, I'm pretty sure, of beta oxidation from the excessive acetyl CoA which was made. And the reason why we take out specifically these carbon units via acetyl CoA is because one of the main byproducts of the oxaloacetate is used for gluconeogenesis, but that's something that we can go on another time because I already know that if I go more in depth on that, we'll go into a rabbit hole. Global bioactivation was greatest in, but was not limited to, lipid-associated macrophages, establishing that diverse myeloid classes undergo extensive metabolic priming in obese adipose tissue. Experimental energetic profiling confirmed the higher basal activity and glycolytic capacity of lipid-associated macrophages over TRMS, sustaining our transcriptome-based flux results. Weight loss led to marked reductions in myeloid cell numbers. And so what are myeloid cells? In hematopoiesis, oh my gosh, forgive me for in hematopoiesis, myeloid cells or myelogenous cells are blood cells that rise from progenitor cell for granulocytes, monocytes, enterocytes, or podocytes. I know progenitor cells are what help build the extracellular matrix and is vital when it comes to protection of the cells. Or in a narrow sense, also often used specifically from lineage. What's its function though? Myelocytes play a crucial role in the immune system, including pathogen recognition, inflammation response, and regulation of adaptive immunity. Oh, I remember these cells. Okay. Myeloid cells are a diverse group of blood cells that originate from the bone marrow. As we know, we have monocytes that originate from bone marrow. They include various types such as monocytes. Okay, so myeloid cells are the class of by which are subclasses monocytes, macrophages, and these protectors of our immune system via inflammation, or can be pro-inflammatory in some cases, such as our lipid-associated macrophages. Hey. So proportional intensity analysis showed that myeloid cell fractions did not differ between obesity and weight loss, and we verified this in situ. Weight loss did, however, shift lipid-associated macrophages towards less inflammatory subtypes. Overall, this indicated that obesity-induced myeloid cell states persist despite extensive weight loss. Transcriptomic flux analysis confirmed that global metabolic activation did not fully reverse with weight loss, but weight loss did significantly reduce some aspects of fatty acid synthesis and oxidation, mainly desaturasis and acyl-CoA synthetases. So that's what I was talking about with acetyl-CoA. We don't have oxaloacetate. We have, from what I remember, and we're not going to go into today, but acetyl-CoA instead. So acetyl-CoA via an enzyme gets converted to acyl-CoA or acyl-CoA, and so that's when it's able to go in the mitochondria and go through the Krebs cycle. However, and then actually then becomes back to acetyl-CoA and goes through beta oxidation, I'm pretty sure. It all meshes together once you try to memorize it all, but we'll do it one day. Temporarily linking these pathways to microenvironmental lipid availability. By contrast, glycolysis, a respiratory capacity, and pentose phosphate pathway flux increased, implying a need to requisition energy from other sources as fatty acid levels diminish. So that just means, by contrast, glycolysis. So as we're seeing here, before we go on further, we're noticing that even with subcutaneous weight loss, even with a drastic weight loss, we are still keeping what seems like the content of these macrophages. However, the difference though is that these macrophages don't necessarily lose in size via proliferation, but they change in roles via differentiation. So we see that while these macrophages may not change specifically in size and number, their role is in their inflammatory response changes. So instead of becoming pro-inflammatory, they start becoming anti-inflammatory. And then we see here is also that while in terms of obese people that are using these fatty catalytic and anabolistic processes are further going into using, for example, glycolysis, which requires glucose in this case to be broken down. And so it's requiring a more diverse use of production of ATP than simply just fatty acid oxidation. Okay. Differential expression and demonstrated widespread reductions in inflammasome, pro-inflammatory cytokine, and chemotaxis genes. Network and analysis implicated specific transcription factors, TFs, and TRM and LAM specification and reviewed patterns reinforcing that the finding that weight loss improves inflammatory and hemostatic networks, but not lipid-associated macrophage transcriptional reprogramming. Together, these results demonstrate a complex activation response in obese adipose tissue dominated by monocyte recruitment and persistent metabolic reprogramming. Okay, so maybe I read that wrong. From what I took of it, I assumed that our lipid-associated macrophages changes and reprograms to be more anti-inflammatory. Now, it seems like, based on reading this, that we are having a reduction of pro-inflammatory cytokines and reductions in inflammasomes, and instead getting more anti-inflammatory cytokines and we're becoming less inflamed. However, it seems like, based on what it finished with the end, that we may actually still have lipid-associated macrophages that are fighting against this homeostatic change, right? And so in obesity, myeloid cells, 31% of immune cells and fats, after weight loss drops to 18%. So overall, immune infiltration goes down, but the fractions of different macrophages subclasses stay similar. Weight loss doesn't erase the obesity-induced composition, it just reduces the total number. Step 2, effects on... So weight loss shifts lipid-associated macrophages toward less inflammatory subtypes. Okay, so I was right about that. This means the balance between adaptive cleanup and inflammatory damaging macrophages improves. However, the obesity-trained state of lipid-associated macrophages doesn't fully go away. Even after weight loss, their memory of metabolic activation persists. Okay, so while it seems like there's a shift within the roles of lipid-associated macrophages when it comes to inflammation response, its inflammatory response, it still has an attack towards our metabolic system, reprogramming. Transcriptomic flux analysis, looking at pathways, not just genes, showed not just fully reversed, global metabolic activation macrophages persists after weight loss. Reduced fatty acid synthesis enzymes, the esterase acyl-CoA synthetases, suggest less lipid availability drives down fat metabolism. So as we talked about earlier, we're using glycolysis and using more of, I'm assuming also probably the Krebs cycle in this case. So in this case, as we talked about earlier, we're using more oxidative phosphorylation we're going to go through not only simply just beta oxidation, but we're also going to go through the glycolysis, the Krebs cycle, and electron transport chain. Right? Glycolysis, glucose breakdown, respiratory capacity, oxidative phosphorylation, pentose phosphate pathway, PPP for NADPH or nucleotide synthesis, implies macrophages rewire to use other energy sources when fatty acids decrease. Okay, step 4, inflammation and signaling. Weight loss significantly reduces inflammasome activation, pro-inflammatory cytokines, chemotaxic genes. So inflammation improves a lot even if the core macrophages intensity doesn't reset completely. Transcription factor networks. Network analysis showed transcriptor factors that govern TRM and LAM identity remain engaged after weight loss, suggests weight loss improves inflammation and restores homeostatic signaling, but doesn't erase the transcriptional programming. So weight loss reduces the number of macrophages, but doesn't fully restore them to lean states. Lean lipid-associated macrophages become less inflammatory, but still carry on obesity memory, metabolism shifts, less fatty acid use, more glucose and PPP use. Inflammation goes down, but macrophage subtype identity remains partially locked in. So it seems like overall that while we're removing that pro-inflammatory response, the macrophages still have that core memory of being able to go back to that attacking basically our metabolic and cardiovascular health, which just makes me curious as to think if maybe this connects as to people who were previously obese have an easier time becoming obese again because of that poor memory within the macrophages still being there of attacking the metabolic system. This suggests that obesity imprints a lasting epigenetic program in macrophages, a kind of metabolic memory. Even after fat is lost, macrophages don't completely reprogram. They still behave differently than in never obese tissue. Potential interventions, epigenetic reprogramming drugs. So it seems like while also when it comes to comparing macrophages on lean tissue in comparison to fatty tissue, they their roles and how they react is still going to be different compared to if someone was of leaner body mass. So low overall numbers, 6,222 cells extended, meant that we were unable to evaluate lymphoid subclass level variations. Nevertheless, obese adipose tissue had higher proportions of CD4 plus and CD8 T cells, NK cells, and B cells. Remodeling effects ameliorated by weight loss. Weight loss also downregulated the lymphocyte activation and cytotoxicity of genes, enhanced adipocyte metabolic flexibility. Mature adipocytes undergo profound phenotypic changes in obesity and weight loss, expanding and shrinking to fit evolving energy needs. How this affects their molecular characteristics and diverse metabolic functions is largely unclear. Subclustering revealed 8 mature adipocyte subpopulations. 2 subtype exhibited stressed and fibrotic profiles. Stressed and fibrotic adipocytes increased with obesity, indicating that there is a selective vulnerability and pathogenicity to the tissue's microenvironment. So we know that fibrosis is via the production of scar tissue, and so you get this via inflammation, right? And so these adipocytes increased. So specifically, these stress and fibrotic adipocytes increase with obesity, which makes sense via the increase of adipose tissue. It's going to increase. You're not going to get stronger. Another subpopulation with lipid biosynthetic profile unexpectedly decreased in obesity. What? Weight loss led to a marked reduction in stress adipocytes, a shift towards lower fibrotic numbers, and relatively increases in lipid biosynthetic cells. Compositional changes in stress and lipid biosynthetic populations were verified in situ. Beige adipocytes were rare. and invariant between conditions. So another subpopulation with lipid biosynthetic profile, so it decreased in obesity. That's honestly, I can see why that's so surprising. Oh, yeah, I don't know why it decreased with obesity, but it makes sense that weight loss led to a marked reduction in stressed adipocytes. Makes sense. A shift towards lower fibrotic numbers makes sense. Less inflammation and relative increases in lipid biosynthetic cells. Personal changes in stress lipo were verified. Okay, so let's break this down though. Why not? So adipocytes in obesity, they expand hypertrophy to store more fat. Okay. So with adipocytes, they expand via hypertrophy to store more fat. Makes sense. And adipocytes in weight loss, they shrink lipid depletion, because we know with in terms of our adipocytes that we have lipid droplet units, and so they shrink lipid depletion. What isn't fully understood, how these size changes translate into molecular identity and function. Okay, so weight loss dramatically reduces stress adipocytes. Weight loss reduces fibrotic adipocytes. Yep. Weight loss increases lipid biosynthetic adipocytes. These come back after weight loss, restoring a healthier lipid handling capacity. Okay, that's cool. What is lipid biosynthetic adipocytes? Lipid biosynthetic adipocytes are specialized cells responsible for the storage and synthesis of fats in the body. They play a crucial role in energy metabolism by converting dietary fats into triglycerides, which are then stored into adipocytes. The process of adipogenesis involves the transformation of precursor cells into mature adipocytes, where they accumulate lipid droplet and become functional fat cells. This process is regulated by various molecular signals and transcription factors, which guide the expression of genes necessary for lipid accumulation and metabolic functionality. Okay, so fat tissue isn't one uniform cell type. We got that. In obesity, more stress, inflammatory, and fibrotic adipocytes, fewer lipid synthesizing ones. However, what we want is those lipid synthesizing cells. So we want these lipid biosynthetic adipocytes. And so while they decrease over increases actually weight loss. I thought it decreases at first. No, increases during weight loss, which is good. So it seems like, as you would assume, decrease of obese adipose tissue will increase healthy I guess biosynthetic. So just like as you would assume, decrease in adipose tissue, increase in lean body mass is overall going to have a better response for your metabolic health and your just overall health in general. Expression-based metabolic flux analysis detected significant defects in fatty acids and branched amino acids. Breakdown in obese compared with lean adipocytes mirrored previous results, and together suggesting that metabolic flexibility was impaired. By contrast, weight loss led to a marked global increase in adipocyte metabolic flux, probably reflecting a negative energy balance. Unexpected anabolic activity led us to investigate whether triglyceride mobilization, which is psychological response to caloric restriction, might initiate lipid cycling, repetitive degradation, and resynthesis. To verify flux models, we compared enzymatic activity scores and pathway limiting enzymes and important substrate pathway across groups. Obese adipocytes had consistently lower metabolic activities scores and enzymes, again indicating metabolic inflexibility. Weight loss systematically increased opposing lipid biosynthesis and breakdown pathways. Consistent with this, we found okay, so obese adipocytes had consistently lower metabolic activities, again indicating metabolic inflexibility. Remember, metabolic inflexibility is the ability to use different energy systems, such as glycolysis and whatnot, instead of just using like beta oxidation and fat energy consuming energy systems. Consistent with this, we found significant changes in chemical enzymes and sequential cycle steps, including DGA2, which encodes an acetyltransferase that catalyzes triglyceride synthesis and mediates lipid cycling in vitro. Because enzymatic expression is crucial determinant of catalytic competence, this indicates that weight loss may initiate triglyceride cycling, a high bioenergetic process with important lipid diversifying, toxic fatty acid quenching metabolic benefits. Weight loss also reversed defects in BCAA metabolism, pathway flux in chemical enzymes, and the predicted consequences of which are systemic BCAA clearance and improved insulin sensitivity. Lipid cycling was a featuring of P and P3 high adipocytes, whereas stress 83 adipocytes were characterized by lower metabolic turnover. These typical catabolic and previously unrecognized anabolic effects of weight loss suggest that substrate mobilization engages cell autonomous cycling pathways that may underlie widespread improvements in metabolic homeostasis. So, BCAA catabolism type of amino acid produ- breaking down of BCAAs. Weight loss improved with breaking down BCAAs, which is important. Obviously, we have amino acids which are vital when it comes to protein synthesis. We talked about earlier via DNA, RNA, mRNA, protein synthesis, production of. So obviously, weight loss weight loss reversed the effects that came with reverse defects of obesity of some obese. So clearly, weight loss has come with positive effects when it comes to using different, having more flexible. So obviously, weight loss has led to some positive effects when it comes to metabolic flexibility, as well as digestion of amino acids. To see which TFs were explicitly responsible for weight loss-induced metabolic activation, we carried out network analysis limited to metabolic pathway genes. MLX ILP and SREBF1 ranked highly in triglyceride synthesis, validating our approach and implicating them in control of weight loss-induced lipid cycling. Other notable findings were TNFs linked to redox biology and BCA catabolism. Many of the leading TFs overlapped human metabolic disease genome-wide association study, casually implicating specific TFs and the respective metabolic pathways and pathophysiology. Casually implicating these TFs and the respective metabolic pathways in pathophysiology and treatment response. So TFs in this case are going to be considered as transcription factors which are going to be important for adipocytes rewiring of our metabolic health. Differential expression analysis identified altered biomechanics as a potential driver of adipocyte stress and metabolic dysfunction that was mitigated by weight loss. Specifically, obesity increased and weight loss decreased expression of key cytoskeletal tension, mechanical transduction, extracellular matrix formation, and fibrosis genes. Effects we verified unbiased pathway analysis and in situ. So altered biomechanics had an effect upon mechanical. That's interesting. Okay. Effects we verified in unbiased pathway analysis in situ. Biomechanical genes were enriched in stress and fibrotic 83 and 86 cells. We therefore evaluated whether adipocyte hypertrophy and a mechanical strain might mitigate these maladaptive changes, and whether adipocyte shrinkage during weight loss might reverse them. As expected, adipocyte sizes increased in obesity and reduced with weight loss. Despite intersample heterogeneity, adipocyte size correlated positively with mechanical sensitivity, stress, and fibrotic gene expression negatively with homeostatic genes. The levels of correlation indicate that this may be one of the several factors eliciting tissue stress and fibrosis, or perhaps it is as a driver event in degenerative cycle. So I'm assuming that. Okay, so let's break this down a little. What do we know about mechanical tension and metabolic stress, and in fact, mechanical stress and metabolic stress? We know that metabolic stress comes along with metabolite buildup via osmosis, in which the extracellular matrix is expanded, enlarged, and this causes stress via the production of growth factors. We know the same thing goes with mechanical stress. However, you're dealing damage via the stretch, cytoskeleton, outside in. You're attacking the extracellular matrix and causing growth factors to occur, improving mitochondrial biogenesis, improving growth, and I think some would argue it actually elasticity of the cell. I was talking with one of my friends' friends upon the extracellular matrix, and so while people may argue that you want to so when people may argue that you may want to increase flexibility of the extracellular matrix, he was arguing that there's actually been some relevant or recent study that might even argue that there's too much flexibility in the extracellular matrix that can lead to a lack of elasticity, which would be a lack of kind of reproduction, I would assume, of recovery of that growth, or in this case would be recovery via from the stretch. It can cause issues in terms of atrophy. However, we'll go on that maybe some other time. However, we know that specifically when the extracellular matrix gets damaged, it calls for growth factors. Same thing when it comes to adipose tissue. We know that adipose tissue can maintain homeostasis via these adipocytes, via macrophages, whatnot. However, what's interesting about this is that these adipose tissues, these adipocytes are able to increase in size. And so as they increase in size, just like with mechanical tension and metabolic stress, they damage the extracellular matrix. However, instead of it increase in muscle growth like you would see via mechanical stress and metabolic stress, this damage to the extracellular matrix makes the extracellular matrix more stiff, less flexible, less, I guess you would say, less adaptive, and making in this case the tissue more fibrotic. The reason being is that instead of it bringing growth factors, it brings fibrotic genes. And so it's very interesting to see how the use of hypertrophy is very similar when it comes to adipocytes in comparison to muscle cells, in that they both damage the extracellular matrix, but the response I get via growth factors or fibrotic genes has an effect upon the extracellular matrix's response to either becoming more flexible or more stiff. And that can connect us with, let's say, sarcopenia. However, sarcopenia is more of a lack of proliferation, differentiation of cells than the extracellular matrix, but there's still a connection which the more that you have a stiff extracellular matrix, the less flexible it's able to come, which means less mechanical tension, which would also relate to having less of a hypertrophic response via less of growth of muscle, having reversal effects of instead of growth, atrophy. Reversal of multicellular stress. Adipocyte progenitor cells, APCs, regenerate mature adipocytes and maintain tissue stroma, crucial homeostatic functions that may become impaired in obesity. APCs clustered into multipotent DPP4 CD55 progenitors, ASC APC1 committed pre-adipocytes, expressing chemical differentiation genes, adipogenesis regulatory cells, and profibrotic precursors. APC3 exhibited a stress profile similar to that observed in mature adipocytes, as well as higher expression of NOCT, a potentially restrictive gatekeeper to pre-adipocyte commitment. In support of this, APC2 selectively expressed late-stage adipocyte maturation genes within a localized subregion. Stressed and profibrotic cell numbers again reflected adiposity and reduced significantly with weight loss. Both populations had higher expressions of hypoxia-inducible factor 1A, which promotes fibrosis and suppresses adipogenesis in mice. Correspondingly, weight loss downregulated hypoxia, profibrotic, and anti-adipogenic genes. Thus, weight loss may attenuate hypoxia-induced impairment of differentiation competency, and prophylactic signaling in certain human APC subpopulations. Coordinated growth of vascular network is essential for healthy adipose tissue expansion. Vascular cell subclustering recapitulated the endothelial arterial. So, as we look here, adipocyte progenitor cells regenerate mature adipocytes. We know that progenitor cells are specifically held within the extracellular matrix, and we know that within the extracellular matrix of adipoid. So we know that adipocyte. So we know that adipocyte progenitor cells. The progenitor cells are generally within the extracellular matrix, and so they help with regeneration and maturation of these adipocytes and maintaining homeostasis. And we know via and so we can also conclude from reading this is that not only are we able to maintain homeostasis, but we're going to get profibrotic precursors via increased fibrosis. But however, as we read this, stress and profibrotic cell numbers again reflected adiposity and reduced significantly with weight loss. So over time, as you lose weight, it's going to reduce this fibrosis. These progenitor cells that are fibrosis are going to decrease over time with weight loss. However, both populations had higher expressions of hypoxia-inducible factor 1A. And so hypoxia is the lack of oxygen. And we need oxygen within the mitochondria in order for us to perform the Krebs cycle and electron transport chain. So with hypoxia, lack of oxygen within the mitochondria is not good, because then this will have us rely on other energy systems which relates back to, for example, excess beta oxidation. When it comes to obese people, we're going to be having a lack of metabolic health when it comes to being obese. Thus, weight loss may attenuate hypoxia-induced impairment of differentiation competency and profibrotic signaling in certain human APC subpopulations. Okay, so with weight loss, hypoxia may be decreased over time, which is great. And as we can tell earlier, is that we're able to use other energy systems besides, for example, beta oxidation and improve metabolic health. Coordinated growth of vascular network is essential for healthy adipose tissue Coordinated growth of vascular network is essential for healthy adipose tissue expansion. Vascular cell subclustering recapitulated the endothelial, arterial, capillary, and venous and mural smooth muscle and parasite zonations observed in other tissue types. Extended as with mature adipocytes and APCs, capillary endothelia and mural cells each showed basal and stress profiles, which change. And so basal is also a part of, we have basal cells, which are key cells in terms of protection, I'm pretty sure, when it comes to our extracellular matrix, which changed reciprocally with adiposity. Stressed endothelia overexpress APOD1 and SNAI1, highlighting potential pathological neovascularization and endothelial to mesenchymal transition. Stress mural enriched to ADAM-TS1, an anti-angiogenic protein linked to parasite detachment, fibrotic transition, and capillary rarefaction. In distinct single nucleus and spatial datasets, weight loss marked reduced stress vascular cell content and markers, implying reversal of this pathological transformation. All stress cell states upregulated a common gene signature. Multicellular stress, although highest in obesity, was a feature of lean tissue, where it increased with age and metabolic dysfunction. Gene and pathway analysis reviewed putative mediators of multicellular stress: hypoxia, mechanical and oxidative stress, mediated cytokines, DNA damage, and cell cycle arrest. In vitro induction of DNA damage using Adipose recapitulated the in vivo effects on stress marker proteins and impaired ASPC differentiation capacity. Weight loss led to a marked reduction in multicellular stress genes, overall emphasizing the importance of multicellular stress pathways in tissue injury and repair. Altered tissue niches and cell crosstalk. We used our spatial datasets to investigate the orientation and impact of stress cells and tissues to define the cells most associated with stress signals. We quantified the cellular composition of low and high stress regions. Stress cell states were generally enriched in high stress zonations, apart from stress capillaries, which were spread throughout the tissue. We also found a strong association between regions of stress and immune cells, except TRM and NK cells, and an unexpected connection to arterial extracellular matrices, ECs. Although this localized individual cell states to stress zonations, it did not address the non-random grouping of cells in microenvironmental compartments. To evaluate this, we used spatially involved proximity enrichment within 300 to capture 300, I don't know what means, to capture adipocytes to search for tissue niches based on the cell state neighborhoods. This identified 5 distinct cellular communities termed arterial, venous, adipocyte, stem, and stress niches. No cell type was niche exclusive, indicating that these patterns reflect tissue gradations. Stem niches were enriched for multipotent ASC, APC1, and homeostatic TRMs. Stress niches were enriched for A3, APC3, LAMS, other innate, C mono, and CDC2, and adaptive T cell immune cells, implicating these states in stress induction and/or response. Arterial endothelial cells formed their own niche, associating with stress precursors, APC3, and stressed mural cells, MU4. Direct cell localization uncovered immune cell proximity to large venous vessels and LAMS, lipid-associated macrophages, potentially reflecting extravasation and transmigration to CLS. Okay, so what can we get out of this? What we can get out of this is understanding that specifically, so what can we get out of this? What we can get out of this is know that cells have niches for stress specifically, and that these cells via stress aren't just scattered around everywhere within the tissue. However, it actually has specific areas by which the cells tend to accumulate. So if you look at here, stress cell states were generally enriched in high stress zonations. So there's zones for these stress cell states, and there's not a specific. So they're not scattered around. They're grouped up, and they're closed up in a specific area. But however, the stress capillaries were the only ones which spread throughout the whole tissue and didn't seem to have any sort of zonation, unlike the rest of the cell states. So we also found a strong association between regions of stress and immune cells, except TRM and NK cells, and unexpected connection to arterial extracellular matrices. So although this localized individual cell states to stress zonations, it did not address the non-random grouping of cells in microventricular compartments. And so they identified 5 distinct cellular communities, and these ones were termed arterial, venous, adipocyte, stem, and stress niches. And so while there was niches that these cells went up to, so while although these stress cells had niches, they weren't exclusive to a particular niche. In fact, it shows right here, no cell type was niche exclusive, indicating that these patterns reflect tissue gradients. So these cells can actually, through gradients, can go into a different niche. And while there are niches in groups of clumped up stress cells, they aren't strictly regulated towards one niche. And so arterial endothelial cells form their own niche, associating with stress precursors and stress myrtle cells. So it seems like specifically was it, yeah, stress capillaries were spread out throughout the tissue. And so same thing is here as well. Arterial endothelial cells form their own niche. So they form their own niche. So while they're able to form their own niche, it doesn't seem like they're technically subjected towards that niche because it seems like it's moved through a gradient. The identification of tissue zonations enables us to investigate intra and inter-niche signaling patterns. Ligand receptor inference analysis in the spatial dataset revealed a complex network of communications. Adipokines and neurotrophic factors were enriched in the adipocyte niche. Mechanical WNT and extracellular matrix components were prominent components of the stem niche. The stress and arterial niches were enriched for pro-inflammatory chemokines. And so it seems like these niches too, as you would assume, have specific functions, right? Mechanical WNT and ECM components were prominent components of the stem niche, right? So in a specific niche, and these stress and arterial niches were enriched for pro-inflammatory chemokines, which makes sense. They're the niches that are subjected for either pro-inflammatory, anti-inflammatory. These specific zones have certain jobs, which is just me basically repeating with what they said. But hey, whatever. Several of which overlapped. Parallel intercellular communication analysis in the larger single nucleus dataset linked diverse niche signals to source target cells, as was disease pathobiology. For example, THBS1, which are emergent triggers of insulin resistance, immune glycolytic metabolism, and inflammation, were all amplified in obesity and have reversed by weight loss. They show that stress niches have a high concentration of signals implicated in pathological and restorative tissue remodeling. So it shows here, we can read here, for example, THBS1, stressed by 83. So these trigger insulin resistance, right? And so this triggers insulin resistance, so this triggers insulin resistance, immune glycolytic metabolism, and inflammation. So THBS1 that triggers these bad metabolic responses got reversed through weight loss. And this shows that stress niches decrease they increase specifically via obesity, but they decrease also as well when it comes to weight loss. Repression of senescence. Differential expression analysis to define adipose tissue remodeling pathways established by that weight loss broadly reverses the effects of obesity on gene regulation. Many of the strongest transcriptional changes associated with weight loss were conserved across several types, indicating that these genes and their underlying pathways might represent important weight loss mechanisms. Genes altered by weight loss in multiple cell types, 3 or more cell types, showed systematic downregulation. Downregulated genes were grouped in hallmark pathways of adipose tissue dysfunction, inflammation. Weight loss also led to downregulation of cell cycle arrest genes, together indicating that the reversal of cellular senescence might underlie the beneficial effects of weight loss on inflammation and metabolism. So it seems here that weight loss causes senescence to these pro-inflammatory cells. Remember, senescence is kind of like the lack of proliferation, differentiation, so the lack of being able to increase in size, but also being able to have tasks in which We know that with adipocytes and its macrophages, and it tracks pro-inflammatory chemokines and cytokines. And so weight loss downregulates the cell cycle and reduces these cells from proliferating and differentiating, and causes reversal of cellular senescence, or causes senescence. And so these will have a beneficial effect on inflammation and metabolism. To examine this, we tested and confirmed the repression of diverse senescent signatures in multiple cell types. Weight loss led to downregulation of CDK and 1A, which is one of the main cell cycle inhibitors in senescence, and the upregulation of cell cycle progression genes repressed by P21. Correspondingly, weight loss marked. To examine this, we tested and confirmed the repression of diverse senescent signatures. In multiple cell types, weight loss led to downregulation of CDK and 1A, which is one of the main cell cycle inhibitors in senescence, and the upregulation of cell cycle progression genes repressed by P21. Correspondingly, weight loss markedly decreased the expression of principal senescence markers and unbiased senescence scores. We found that P21 positive cells, which had transcriptional characteristics of senescent cells, were most prevalent among stress adipocyte, precursor, and vascular cell states, indicating that the shared stress profile reflects vulnerability and transition to senescence. Lean adipose tissue also contains substantial numbers of P21 positive cells. By contrast, weight loss completely eliminated P21 positive cells from the tissue, a finding that we verified in situ using spatial transcriptomics and protein quantification. The repression of senescence mirrored enhanced adipocyte bioenergetics, indicating that these effects may be mechanistically coupled. We therefore established that human weight loss has previously undescribed potent senolytic effects. Tissue-wide gene regulatory network analysis revealed a tightly conserved transcriptional nexus in stressed senescent cells that increased in obesity and decreased in weight loss. They identified TFS group in several classes. The AP1 superfamily, which primes the senescent genome, hallmark signal-dependent TFs that activate inflammation in the senescence-associated secretory phenotype, which are implicated in cell cycle arrest. TFs that control cellulogenesis. What is cellulogenesis? Cellulogenesis is defined as the building of the cell's antenna, or the extracellular fluid mediation mechanism. It's motility. It's movement. It includes the assembly and disassembly of the cilia during the cell cycle. So what is? Let me just make sure I have the conditional process of deterioration with age. Leaves. Okay. So in this case, maybe I got it wrong, but I'm pretty sure. Okay. So senescent cells increased in obesity but decreased in weight loss. So, which makes sense, but I think I might have reversed of what I was talking about then. I thought that there was a senescence within the adipocytes in specific. I think that was the case. Okay, yeah. So weight loss led to downregulation of CDK and 1A, which is one of the main cell cycle inhibitors in senescence. So it actually seems like I got it wrong. It seems like with the increase of adipocytes via obesity, we have an increase of senescence within our stem cells, right? And so what's cool is that weight loss actually led to downregulation of CDK and 1A, which is one of the main cell cycle inhibitors in senescence. No, I was right because it inhibits senescence. Let's get this chat GPT. Let's figure this out. Okay. Senescence markers. Weight loss downregulated CDK and 1A, P21, a chemical marker of cell cycle arrest and senescence. Weight loss also decreased global senescence scores. So, weight loss is not causing senescence in pro-inflammatory cells, it's doing the opposite. It reduces stress burden and either prevents stress cells from transitioning into senescence or selectively eliminates already senescent cells. Okay, so we see here that weight loss downregulated CDK and 1A, which is a chemical marker of a cell cycle arrest and senescence. So these P21 cells increase senescence, which is the lack of proliferation differentiation, which is not good at all. And so this will, and due to the lack of proliferation differentiation of other cells, we have enriched stress in adipocytes, precursors, APC. So this tissue-wide gene regulatory network analysis reviewed a tightly conserved transcriptional nexus in stress senescent cells that increase in obesity and decrease in weight loss. The identified TF group grouped into several classes: the AP1 superfamily, which primes senescence genome, hallmark signal-dependent TFs that activate inflammation and senescence-associated secretory phenotype, purple-like TFs, which are implicated in the cell cycle arrest, PFs that control sialogenesis, which is a putative synthesis regulator, orphan nuclear receptor TF. So we read this. Okay. So ignore the passing what I just said. Okay. Because of its importance in reinforcing senescence, we ought to further define the signature of the AT-SASP by systematically comparing the expression of secretory proteins across high senescence and basal cell states. This revealed changes in diverse mediators of senescence, tissue injury, and metabolic dysfunction, including signaling peptides implicated in multicellular stress and intra and interniche communication. Resumptive SASP components systematically replicated in situ and localized to stress and arterial niches. Senescent cells strongly upregulate NAPT, an intracellular driver of SASP, and an extracellular adipose cytokine and pleiotropic context-dependent, predominantly pro-inflammatory effects. NAMT expression was similarly enriched in obese macrophages and inflammatory lipid adipose macrophages, in keeping with its roles in inflammasome activation and immune recruitment. Tissue-level protein analysis confirmed that NAMT abundance increased in obesity and reduced markedly with weight loss, together highlighting that NAMT is a likely driver of AT-CSP. In summary, these analyses reviewed diverse intracellular and extracellular mediators of degenerative adipose tissue senescence cycle and support reversal of adipose tissue senescence as a key determinant of metabolic health benefits of weight loss. As the number of people living with obesity surpasses 1 billion, there has never been a greater need to understand the opposing effects of obesity and weight loss on metabolic health. Here, we report a high-resolution signal nucleus in a spatial atlas of human adipose tissue in people with extreme obesity undergoing therapeutic weight loss and a healthy lean counterparts. The simultaneous analysis of obesity and weight loss enables us to understand the core tissue remodeling principles, capture more than 20 cell states that vary with body weight, including degenerative and adaptive populations that bridge cell ontologies, and define molecular pathways, regulatory factors, and intercellular signals that may drive tissue injury and subsequent recovery. Foremost, we review selective susceptibility to cellular stress and senescence in subpopulations of metabolic precursor and vascular cells, but not in their immune counterparts. This susceptibility is amplified in obesity, but is evident in aging and metabolically unhealthy leanness. We discovered that weight loss has potent mitigating effects on senescence and vulnerable cell types. We predict from transcriptional patterns and previous mechanistic studies that leads to increased metabolic flexibility in mature adipocytes, improved differentiation capacity in precursors, and recovery of vascular abnormalities. Deep molecular phenotyping across cell types, cohorts, and modalities enables us to define a tightly conserved neuroregulatory network that may elicit and reinforce human adipose tissue senescence. Putative upstream triggers key components of degenerative AT-SASP vulnerabilities such as MCL1 that might be exploited with therapy. We conclude that with the reversal of adipose tissue senescence may be central to the multi-organ and anti-inflammatory and metabolic benefits of human weight loss. So you can see here, as we decrease this senescence, let's say P21, we increase our anti-inflammatory and metabolic responses via weight loss, right? So, which is great. By modeling enzymatic gene expression, we show that weight loss induces global metabolic activation of mature adipocytes, presumably to release stored fuel to meet energy demands. Two prominent activated pathways are BCAA metabolism and lipid cycling. Contrary to expectation, both pathways are bioenergetic, challenging the assumption that weight loss reduces energy expenditure in AT, suggesting that there is compensation elsewhere and warranting further investigation. Irrespective of this, we propose that pervasive activation of substrate turnover in adipocytes has effects on insulin sensitivity and ectopic lipid that may be crucial in the multi-organ metabolic benefits of human weight loss. Despite these effects, our lean cohort was the healthiest, indicating that other factors are involved. In the immune compartment, we confirmed that the human obesity leads to monocyte and macrophage infiltration activation to a lean adipose macrophage phenotype. We extend this lipid adipose macrophage phenotype. We extend this lipid adipose tissue macrophage phenotype to metabolic dysfunction independent of body weight and uncover gene regulatory mechanisms and metabolic pathways implicated in LAM specification activation. Consistent with an adaptive maladaptive spectrum, we find that LMS exists on a continuum. At one extreme, we define a TLR2 TREM1 inflammatory LIM signature that associates with adiposity and metabolic dysfunction, akin to protherosclerotic LAM subtype. We show that after a weight loss that there are marked reductions in monocyte and macrophage and lymphocyte inflammation pathways, which we predict even in the absence of classical activation to be anti-inflammatory. Despite overall reductions, we observe persistence of obesity-induced macrophage activation states that are probably epigenetically programmed. In mice, AT immune and metabolic cell memory is implicated in weight gain, regain, and enhanced inflammation. Thus, persistent macrophage activation in human adipose tissue may impede complete metabolic recovery, trigger weight regain, and worsen long-term clinical outcomes. So it's interesting, and to break it down simply, it seems like these macrophages, as we explained earlier, are still have a programming to go back to their state at which they were at when they had a lot of, for the people being tested that had weight loss, to go back to the point at which they were at their obese state, right? And what that means is that when that they're okay. So simply, if we break this down, it seems like these macrophages want to go back to its inflammatory state. What I mean by that is that people that people who were at an obese point and decreased via weight loss, we talked about earlier that they're programming via the macrophages, while macrophages went from pro-inflammatory to anti-inflammatory, still have that coding inside their system that allows them to basically memorize and go back to that pro-inflammatory, poor metabolic health state. And it seems like in this situation that people that have undergone weight loss, originally being obese, have a high susceptibility to having these issues of poor metabolic health and pro-inflammatory macrophage activation. We see here in mice, AT immune and metabolic cell memory is implicated in weight regain and enhanced inflammation. And so it seems like with people that have had an issue in history of weight problems, have a higher susceptibility of having drawbacks in the later future of going, one, having weight gain, and two, having more health problems that was very similar to where they're at when they were at their obese state. So thus, persistent macrophage activation in human adipose tissue may impede complete metabolic recovery, trigger weight regain, so it may cause more metabolic health issues, trigger weight regain back, and worsen long-term clinical outcomes. So it's very scary to imagine that even though you have lost weight, people that have had issues in the past with their weight are more susceptible to having health problems, even if they're at a healthier state. We studied weight loss in its early phase to define potential driver mechanisms. Because of this, we can unravel the respective contributions of negative energy balance, weight change, and absolute fat mass to observe tissue and systemic effects. Other limitations include a focus on people without diabetes, the abdominal subcutaneous depot, and surgical weight loss, variable biopsy methods, and incomplete capture of rare immune cells. Previous studies indicate that adipose tissue in different locations has important phenotypic differences that may contribute to variability in weight loss outcomes. However, the degree of fat mass loss remains one of the strongest predictors of what the fuck is this? However, the degree of fat mass loss remains one of the strongest predictors of metabolic response, irrespective of intervention. Thus, we anticipate conserved but also context-dependent adaptations across adipose tissue compartments and weight loss methods. Collectively, our results reveal that weight loss has significant effects on cellular processes that are known to affect metabolic health and longevity. More broadly, our findings highlight the need for proactive obesity prevention and support the possibility that sustained lifestyle changes could have long-term health benefits mediated through dynamic remodeling of diverse AT cell types. This rich representation of human AT biology and pathophysiology offers a valuable resource for mechanistic and therapeutic exploration. So here, it shows that obviously, well, not obviously, but it shows that different locations of adipose tissue might have an effect upon phenotypic differences, maybe cellular processes. However, it seems like there's also cells too as well, unfamiliar cells that we weren't able to keep to track. However, it seems like overall the weight loss outcomes tend to be consistent all the way through, generally, when it comes to, let's say, the roles of macrophages changing as you decrease adipose tissue. However, also showing an outcome of less, in this case, what we looked over earlier, an example, is less accumulation of pro-inflammatory adipose cells, such as, okay, methods, studies on. Single nucleus RNA sequencing, snRNA sequencing, was done in subcutaneous abdominal adipose tissue from 25 people with obesity before and after weight loss, paired samples, and in 26 healthy lean controls. 2 lean samples were removed as described below. Obese case and control groups were well matched for age, sex, and ethnicity. Spatial transcriptomics was done in equivalent groups, n equals 4 per condition, as were histological cross-validation studies, n equals 4 to 5 per condition. All molecular phenotyping was done after overnight fasting. The weight loss interval was a minimum of 5 months, median 7, range 5 to 18 months. Median percentage weight loss was 22%, ranging 13 and 33%. Primary snRNA sequencing data was integrated with previously published snRNA sequence data in whole subcutaneous adipose tissue from 9 obese and 4 lean people to increase obese and lean cell numbers and improve cell annotation. Participant characteristics are provided in the extended data table 1. So, what gets me curious is median rate percentage weight loss was 22%. Okay, so this is a range from 13 and 33%. As we looked earlier, as we get closer to becoming obese, and we know that I think obese levels consider anything past 30% body fat percentage. Generally, for a male, it's supposed to be recommended that you're between 14 to 18%, and then beyond 25%, you're considered overweight, and then beyond 30%, you're considered obese. I wonder, with this range ranging from 13 to 33%, if the results of, let's say, when we looked at adipose tissue and its response on one, reliance of beta oxidation, while also having a difference when it comes to certain groups, cell groups that were obviously has gradients in between that can, I guess, go and maybe not maybe it could. And we saw also with this adipose tissue that there were certain groups of cells clumped up within this adipose tissue, so they weren't sporadically everywhere, but they were grouped up. I wonder if the range of 13 33% of body fat is too high of a difference in range when it comes to, one, taking account of the state of cardiovascular health and metabolic health, while also taking account of the fact of how much M2 and M2 macrophages are being accumulated via adipose tissue, while also taking account of, like I said, the reliance on fatty acid oxidation or any sort of fat catabolism or anabolism process, right? So I wonder if, I know the median percentage weight loss was 22%, which is this referring to, maybe it's a loss. Okay, I think it's referring to a loss. I might be totally wrong in this situation. Okay, well, I'm probably wrong in this situation, but it's something to think of when I was reading it. The adipose tissue samples were obtained upwardly from morbidly obese individuals. BMI, okay, so these are people, never mind. So this is the amount of weight loss between 13 and 33%. Okay. The BMI was over 35 kilograms, undergoing laparoscopic bariatric surgery, gastric bypass, or gastric sleeve in healthy controls, undergoing non-bariatric laparoscopic abdominal surgery. Subcutaneous adipose tissue was collected from abdominal surgery incision sites. Follow-up subcutaneous adipose tissue samples were collected from the para-umbilical region using needle biopsy more than 5 months after weight loss intervention. Whole adipose tissue samples were snapped frozen at the collection and stored at negative 80 degrees Celsius for future use. Participants were unrelated, between 20 and 70 years of age, from multi-ethnic background and free from systemic illnesses not related to obesity. So what gets me also curious too is that these are people between the ages of 20 and 70 years of age, right? And so we know that over time, and honestly, I've had two perspectives upon this, is that specifically the extracellular matrix can either stiffen up, right? And so it becomes less flexible over time, and that creates us to have one of the results of atrophy and a lack of muscle growth over time, also with the account of a lack of cytokine production and proliferation, differentiation by cells via growth factors. I've also heard that the lack of muscle growth may be actually because of weakening of the extracellular matrix, making it too flexible for it to kind of regrow again. Think of it as like a band that's super elastic but won't have any power to go back. But what gets me curious is that we were looking at earlier, we're looking at adipose tissue and hypertrophy of adipose tissue. And we're looking at how specifically it gets fibrotic genes to accumulate to the extracellular matrix once it expands in size via hypertrophy, which causes fibrotic scarring, right? And it causes this accumulation of even more inflammation. I wonder if the participants being between the age of 20 to 70 years old, with 20-year-olds having obviously a more healthier response towards damage and cell regrowth. I wonder if the age gap would be too much of a diverse separation. It's too much of a separation in order for it to get an accurate response. But I guess we'll see as we continue on. From a multi-ethnic background and free from systemic illnesses not related to obesity, people with treated type 2 diabetes were excluded because of the potential effects of hypoglycemic medications on adipose tissue metabolism. Metabolic characteristics were collected at baseline follow-up. All participants gave informed consent. The study complies with all relevant ethical regulations. It was approved by the London, City Road, and Hampstead Research Ethics Committee, United Kingdom, reference 13 LO 0477. Human tissue validation also used samples from the Imperial College Healthcare Tissue Bank, approved by Wales REC3 to release human material for research, reference 17.0161. Okay, so now we look at the nucleus isolation. The adipose tissue nuclei from the individual participants were pooled for a library preparation and sequencing to increase efficiency or diversity in study power. Pooled samples were separated by condition to avoid crossover, 4 to 5 samples per pool, a total of 6 pools per group. Sample pools for each experimental group were processed to sequencing in lean, obese to weight loss trios to minimize between group batch effects. For each participant sample, nucleus extraction was done using a modified version of previously described protocol. In brief, frozen human adipose tissue, about 100 milligrams, was cut into pieces less than 0.2 centimeters and homogenized with 1 milliliter ice cold lysis buffer, 0.1% MP40, as well as it being in a glass doused homogenizer on ice. After homogenization, what is a homogenizer? A homogenizer is a device used to break down and evenly distribute particles with liquid mixture, creating a stable and uniform emulsion, suspension, or solution. It works by applying various forces such as turbulence and cavitation, often at high pressure, to ensure a consistent blend. Homogenizers are commonly used in laboratories and industries for fixing, emulsifying, and homogenizing liquids. So I wonder if this homogenizer was helped to use to break down and separate the nucleus away from the cell. That'd be very interesting. I find the process of specifically grabbing the nucleus and using its DNA information, or in this case maybe its RNA information, and how it's sequenced, very interesting when it comes to be able to find all this information. For each participant sample, nucleus extraction was done using a modified version of previously described. Okay, we look through this. Let's go. Okay. After lipid supernatant removal, the nuclei pellet was suspended in 1 milliliter wash buffer, transferred to new 1.5 milliliter low DNA bind tubes, and reintrifuged at 500 grams and 4 degrees Celsius for 5 minutes. After repeat lipid supernatant removal, the nuclei pellet was resuspended in 300 UL wash buffer containing DAPI, thermoscientific 62248. I'm not even going to read this shit. At this point, the isolated nuclei from 4 to 5 samples from the same experimental group were pooled before sorting by flow cytometry. FACS was used to clean up the residual debris and lipid from isolated nuclei to remove doubtlets. Pooled nuclei were sorted on a BD FACS area sort. The sheath tank was bleached cleaned before each run, and nucleus-free PBS was used as a sheath liquid. A 405 nm laser was used to excite DAPI, and emission was collected using 450 to 50 nm bandpass filter. I need to figure out nm. Single nuclei were selected by getting on the first DAPI positive band on the DAPI versus forward scatter plot, and then subsequently gating on side scatter versus FSC and FSCA versus FSCH to ensure better debris and doublet removal. All sorts were performed using an 85-degree nozzle. The sorted nuclei were collected into a BSA and RNS inhibitor-rich collection buffer. In low DNA bind tubes kept at 4 degrees Celsius after sorting, nuclei were centrifuged at 500 grams for 5 minutes at 4 degrees Celsius to pellet. Supernatant was removed to leave about 40 UL, which was used to resuspend pellets with a wide bore pipette tip. Pooled single nucleus suspensions were used to generate barcoded single nucleus libraries for the next generation sequencing. For each pool, 5,000 to 10,000 nuclei were co-encapsulated with a 10-time barcoded gel beads to generate gel beads and emulsion gems using a 10-time chromium controller and a 10-time genomic single cell 3V.31 kit, according to the manufacturer's instructions. After GEM, RT, and cleanup and quantity and fragment size distribution of amplified cDNAs, so remember, cDNAs are copy DNAs derived from barcode single. So what they're doing right here, they're copying this DNA in order for them to just transcribe it. Derived from barcode single cell RNAs were assessed using Agilent 2100 bioanalyzer high sensitivity DNA assay. From this cDNA, snRNA sequencing libraries were reconstructed and sequenced in 3 batches containing equal numbers of obese, lean, and control library pools to minimize between group batch effects. Each unique library was sequenced to a minimum depth of more than 20,000 paired end reads per nucleus. Raw sequencing data were demultiplexed and analyzed using Cell Ranger V.501 and BCL2 Fast V.2.20.0 libraries were demultiplexed using Cell Ranger MK Fast Q based on the sample indices, allowing one mismatch in the Cell Ranger count pipeline was used to perform alignment against the human genome GRCH38 using STAR, filtering and counting unique molecular identifiers. UMIS, including introns. What are introns again? I know the introns are going to. Introns, on the other hand, are non coding sequences that interrupt the coding regions and are removed during RNA splicing before translation. Exons are the coding region of a gene that are expressed to the final messenger. Okay. Because remember, when we have. Let's see. So I think the exon is the. So the exons, I think, are the stop codons. And the introns, on the other hand, are non-coding sequences that interrupt the coding. And so they are removed. Okay. For each pooled library, raw count matrices from Cell Ranger were processed using CellBender. Epochs 150 to 200 learning rate. To remove ambient RNA molecules and random barcode swapping and filter inferred cells. Okay, so we're gonna go forward and go on this if you're really interested. But I want to go into the compositional analysis and the metabolic analysis. Well, yeah. Let's go to the analysis overview. So the analysis overview. Cell type and state annotation was done and combined our own and that from reference integrated dataset. Primary exploratory analysis formed in our own dataset, which was processed in experimental group trials, which was processed in experimental group trios, lean, obese, weight loss. To minimize batch effects and comprise paired obese weight loss samples in age, sex, and ethnicity matched link controls. The referential, neighborhood, abundance, and expression analysis between groups in which biological, technical, and batch covariates could be adjusted for or separated were repeated using the combined dataset to verify reproducibility. We identified the main cell types with unbiased clustering using a low resolution leaded algorithm, and each cell type was annotated according to known markers. To identify cell states, we isolated the barcodes for each of the main cell type identities, except for mast and lymphatic endothelial cells, owing to low numbers. Each cell type was then reintegrated and reclustered twice, as described above. First, we used a high-resolution lead-in 1.2 or higher to identify barcodes that contained a mixture signature with markers of different lineages. These barcodes were flagged as unassigned and were excluded from any downstream analysis. Then, we removed these barcodes and proceeded with the second round of reintegration clustering. Resolution varied across cell types, 0.65 or higher, with myeloid cells requiring the highest lead-in to identify rare known cell types, 2.25. Clusters that were similar to each other had no unique identifiable features that were merged. Cell states were annotated based on a mix between unbiased and known markers. To identify unbiased markers, we used Scampi's rank gene groups to function and perform a Wilcoxon test. Composition analysis. To analyze... Okay, let me see. I want to know... I think what we can get out of this research study is the importance of taking care of your health. Obviously, we saw the adverse effects of obesity, specifically on your metabolic health as well as your cardiovascular health. cardiovascular health, the relationship with your blood vessels and your heart and your metabolic health, being able to create energy. As we saw that people with obesity struggled with going through oxidative phosphorylation via the Krebs cycle, via the electron transport chain. They showed a lot of oxidative stress, lack of mitochondrial biogenesis, and an increased proliferation and differentiation of adipocytes specifically. We saw that with adipocytes, they can go through hypertrophy via breaking the extracellular matrix and making the extracellular matrix more stiff. And so we can also see the relevance and importance of keeping and sustaining a healthy body fat percentage. I think there's an epidemic going around right now, or constantly bulking and cutting. And so, as we saw, people that were at a obese state that went through weight loss were not in the same state via cells, such as their macrophages, in comparison to people that maintained a lean body mass. We saw that specifically macrophages and cells that recruit cytokines and chemokines to the immune system still had that pro-inflammatory coding within their DNA sequencing. And so while they were not pro-inflammatory, while they transferred from becoming M2 fat adipose adipose macrophages, while they converted to more M from what I think M1 macrophages, but basically became anti-inflammatory. While they changed their states of being from pro-inflammatory to anti-inflammatory, we saw looking through the copy RNA that they're still sequenced for those negative adverse effects towards metabolic health. And so people who were at an obese state that went down to a healthy weight had a higher chance of these adverse effects of their macrophages being pro-inflammatory. So just keep this as a sign for people that are obese now and are trying to lose weight, that there were great and positive effects towards inflammation, fibrosis, in which there was a reduction of pro-inflammatory response when they lost weight. However, we can have this as a telltale sign that for people that are fluctuating in weight or thinking about going from having like a massive bulk to a massive cut, that there are significant effects towards your cellular responses via your cells and your DNA sequencing when you are at a point of adipose tissue that's unhealthy. So that's what we're going to get at for today. Hope that was interesting. If not, then that sucks. But yeah, have a good day, guys.